Hello.
You can see.
I hope you can see my slides.
Seeing your slides, proceed, please.
Okay. Anastasia Mawel, a student from Animal Science Department, Egerton University. Kenya. I'm presenting on digestibility and in vitro methane gas emission of indigenous and introduced grasses in the rangeland ecosystem of southeastern Kenya. This work was supervised by Professor Bebe, Dr. Olivia Kashongwe, and Dr. Simon Kuria from Karo. And this is the outline of my presentation. I'll begin with the introduction, statement of the problem, hypothesis, materials and method, conclusion, and recommendation, and finally acknowledgement. As part of the introduction, rangeland ecosystem, they support large proportion of ruminant production. And the most dominant grass species are the indigenous perennial grass species in those rangelands, including Erangrostotis superba, Sengrass ciliaris, etc. And these grasses they form the main source of desert diet for livestock in the rangelands. And these grasses are highly promoted in the Aso regions, but they are scarce in terms of quality and quantity. The predominant grasses, and this makes them fail to support the increased productivity for livestock. And this is from their low biomass yield and their poor quality. And this poor quality is highly associated with high methane emission, particularly from high fiber content. Some tropical improving grasses from high rainfall areas have been introduced in these rangelands to supplement the feed base. But their response to high temperatures is low, hence limiting their performance. Thus, an intervention is highly needed to improve productivity of livestock, and this intervention can be done through improving pasture management so as to meet the rising demand for livestock feed and also mitigate the negative effects of climate change. So in doing this intervention, we need to prioritize not only on feed quantity, but also on the quality of feed, such that we have a potential sustainable ruminant production. But the evidence is scanty on the endemic and methanogenic production potential of the locally available grasses, as well as for the introducing grasses. This study tested the hypothesis on digestibility and methane emission to test the significance between digestibility and methane emission of the introduced grasses and the indigenous grasses. And the result from this study has a co-benefit of, should inform the grasses with co-benefit of high digestibility with high nutritive value and low ederic methane emission for sustainable ruminant production in the rangeland system. The statement of the problem. Livestock production in the Aso is faced with feed scarcity, as I have said, in terms of quality. That is the fact that the feeds in the Azos are highly lignified with high fiber content. And also in terms of quantity, the feed is scarce, is not enough for the livestock. So the indigenous grasses, which are predominant in these Aso areas, are highly promoted to meet the feed base and highly disseminated to farmers to feed the big needs. But their potential to the environment, their potential to increase productivity is low due to their low quality. Also, introducing grasses are being introduced from the high rainfall areas to the rangeland to supplement the feed base. But they are being, the promoted grasses, both the indigenous and the introduced, are continuously being utilized without the good knowledge of their methanogenic potential and their potential in improving livestock productivity. And this will necessitate for the grass pastures with multiple attributes of high biomass yield, high nutritive value, and low methane emission. This study was geared towards a general objective of contributing to sustainable regional pasture production and livestock production and a regional ecosystem. And this was through determining the digestibility and methanogenic potential of the introduced indigenous grasses and the regional ecosystem of southeastern Kenya. And the objective was to answer the hypothesis that digestibility and methanogenic potential is no significant difference between the introduced and the indigenous species. Under materials and method, this study was carried out from Arid and Regional Research Institute in Kiboko, a Kauro station in Kiboko in Makueni County, and the samples were collected at Bloom State. The samples collected were for the 3 indigenous grasses, that is Dangrostotis macrostatus, Sengras iriasis, and Dangrostotis superba, and 2 introducing grasses of Chloris Nkayana variety, that is Vomarods and Extosirods. The samples were dried at 65 degrees Celsius and ground in an oven here at Arinda Regional Research Station and ground 1 millimeter sieve mill. The ground samples were then analyzed for nutritive quality as follows. For dry matter, they were burnt in the oven at 105 degrees Celsius for 24 hours. For us, they were combusted in a maple furnace for 550 degrees Celsius for 4 hours, using the method as in the screen. The total nitrogen content was determined by using the DAL procedure. Crude protein was obtained by multiplying N by a factor of 6.25. Neutral detergent fiber was determined by the method of OSC 1990, method 6.5.1, and acid detergent fiber following the method as in the screen. In vitro digestibility and gas production was done using the method by Menge and Stenikas, as described by Fujihara 1999. During in vitro digestibility, gas samples were collected after 3 hours of intubation from the syringe head space and put in glass vials. Then the collected gases were taken to Iri, where they were quantified for the concentration of methane gas production using gas chromatography. The experimental design used was complete randomized design, and the data was analyzed using SPSS version 22. And the model fitted was as shown on the screen. Then later I ran the simple regression, fitted the linear regression equation shown to determine the association between methane emission and digestibility of the grass pastures. Results and discussion. On chemical composition of experimental grasses. From the study, I concluded that I found out that the crude protein and acid detergent fiber was significant. There was a significant difference between the introduced and the indigenous grasses, with the introduced grasses having the highest average on average, having the highest CP and ADF. While dry matter, neutral distillate fiber, and acid detergent lignin was not significant. The low CP for the indigenous supplied insufficient nitrogen for proliferation of rumen microorganisms. And this led to low fermentation level compared to the introduced grasses. High NDF content in the grass also mean low supply of readily available energy to nourish the microbial fiber, and this also suppressed the activity, hence low indigestibility. The in vitro dry matter digestibility and methylene production values for the studied grasses was found to be significant, with introduced grasses having high digestibility and high methylene production compared to the indigenous grasses. And this can be tied to low CP content for the indigenous grasses and high ADF, acid detergent fiber. The in vitro digestibility of the indigenous grasses could also be, the lower digestibility could also be associated with NDF. From there, we can see that the indigenous produce less methane per gram dry matter compared to the introduced grasses. Looking at the trend, we find that the introduced grasses, that is the Chloris Guyana variety, Extosiros and Bomaros, showed the highest trend in methane production as compared to the indigenous grasses. What is the message from the study? The introduced grasses produce the highest volume of methane compared to the indigenous grasses. And the indigenous grasses showed low CP constraint and high fiber content, which is a limiting factor in animal productivity. So while feeding on livestock for the productivity on the indigenous grasses, we recommend nitrogen supplementation, maybe using leguminous feeds or using concentrates to boost the nitrogen supply for efficient performance of the microbes and productivity. As a conclusion, ruminant production utilizing introducing grasses, I concluded that would achieve increased productivity, but with sustainability trend of increased methane emission, and this is a sustainability concern in the regional ecosystem. I wish to acknowledge Egerton University for organizing this conference and for offering me opportunity to study in the university. CSAM, Center of Excellence in Agriculture and Business Management for funding this study. Cairo for allowing me to use their lab and to collect samples from their plots, really for analysis, and my supervisors for their endless support, resourceful support. Thank you. Thank you.
Thank you so much, Anastasia. Thank you for your presentation. You have done it within time. And may I ask you for questions or clarifications from the audience? I can see one question in the chat. Have you seen it, Anastasia, for you?
Let me see it.
Have you seen it?
Yes, I've seen it.
Yes, can you respond to that one?
Under ingression, I was looking at the association between digestibility and methane emission. So I can say I have the two points as per the hypothesis are taken care of.
Have you, the person, I'm sure, satisfied with your explanation? Because the question is, why is the regression model having one predictor, that is digestibility, as opposed to two, as shown in the null hypothesis? In the null hypothesis, you have shown two, but in the regression model, you have only shown one. So there is a discrepancy. Can you explain that a little further? Anastasia, can you please explain that further?
On the regression model.
Regression model vis-a-vis what you presented in your null hypothesis.
Yeah.
In the hypothesis, you indicated two indicators or predictors, but in the regression model, you have only alluded to one, which is digestibility. So can you explain a little, because it seems that there is a discrepancy.
And then in the null hypothesis, the study was to...
Can you go back to the hypothesis and then you flash back the regression model? Take us back to the hypothesis.
That is what I'm trying to do, but it is not going back.
Stop sharing so that I share it from this end.
Okay.
Or share it afresh, Regina. Sorry, not Regina, Anastasia. Stop sharing and then you start afresh. Are you succeeding, Anastasia?
Yes.
Okay, so can you put back your hypothesis?
Hypothesis, yeah, I'm there. My hypothesis is digestibility and methanogenic potential is no significant difference between introduced and indigenous grasses. And on my regression model, methanogenic potential, which is
Anastasia,
yes.
Kindly share your screen. You're not sharing the screen.
I'm trying to share. Please share from your head. I'm facing some difficulties in sharing.
Okay, let's
okay,
share.
I have succeeded. Is
Okay.
it visible now?
Yes, it is visible.
Okay. Looking at my hypothesis.
Yes, so here, how many predictors do you have?
From my hypothesis, I was looking the association, the significance between digestibility and methanogenic potential between the grasses. But that is the introduced and
yes,
the indigenous grasses. Now looking at
the regression model.
the regression model, the independent variable, which is the methane emission potential, is the methanogenic potential. And the independent is not the digestibility.
Okay.
Yeah.
I don't know whether Dr. Bishori is satisfied with that explanation. If so, then we go to the next person who has a question or a comment to make from.
Maybe, Chair, maybe I can ask.
Yes, please, go ahead, introduce yourself.
Thank you, Chair. My name is Dr. James Moshori from the University of Nairobi and a project management specialist. When you look at the hypothesis, the way it has been stated and the regression model that where we have the predictors that are fitted in, we have the beta 1 predictor X1. So when you look at it down there, the interpretation is digestibility of grass pasture. So where is the other, it was digestibility and something else. So, you know, now you have to be very careful because someone would be asking, why do you have digestibility and then another predictor? So if at all digestibility is part of what the other thing that you have mentioned in your hypothesis, then it has to be full here. But if you leave it digestibility, there will be a question on why the other one appears like a predictor there. So that maybe you need to consider correcting. Thank you, Chair.
Anastasia, have you gotten that point?
Yes, yes.
Are you going to correct that?
Yeah, I look on it.
Okay, thank you. Thank you, Dr. Hari. Any other person with a question or point of observation to improve Anastasia's work? Perhaps as we await for somebody to ask, Anastasia, may I ask you something for clarification?
Okay.
In your conclusion, and if you
Yes.
can put back the conclusion, please, the slide with the conclusion.
Yeah.
So you're saying that introduced grasses
Yeah.
performed the indigenous grasses. Is that so?
Yes.
In terms of what and what?
In terms of the nutritive quality,
Yes,
but in terms of contribution to the environment,
yes.
the indigenous outweighs.
There is also another aspect,
Yeah.
the aspect of durability. The indigenous grasses tend to withstand harsh conditions, especially lack of moisture and soil irrigation and so forth.
Yeah.
So to me, I'm a layman. Let me assume I'm a layman. To me, I would say this: if the indigenous grasses will persist longer and assure some provision of food to the animals,
Yeah.
in this ecosystem that is very fragile, and moreover, they are not contributing so much to global warming, which we really want.
Yeah.
Would you really persuade me to go for the introduced grasses?
Also, I can say the indigenous grasses, since they are durable and highly adapted to climatic change, as from this slide, the indigenous grasses can continue being promoted, but while feeding to the ruminants, we recommend nitrogen supplementation. And this nitrogen supplementation will lower their contribution in terms of methane emission to the environment.
I'm still following up that point, Anastasia, because for ruminant animals, they need very minimum crude protein, isn't it?
Yes.
Just some source of nitrogen to make the rumen microbes happy, and the animal will be happy, and the farmer will be happy. So if these indigenous grasses are going to persist, look, we are looking at your ecosystem is very fragile. When it is very dry, the way it is dry now, there is no sufficient pasture. And if you are looking at Rhodes grass, I am sure if you step out from where you are and you look at a plantation of Rhodes grass compared to star grass and these indigenous grasses, Rhodes grass is no longer there, isn't it?
Yes.
So the trade-off, my question is about trade-off. You are looking at the good. So now the indigenous grasses are going to assure the animal at least something to bite, even if they provide little protein, isn't it?
Yes.
But that little protein is what the rumen microbes need just to make themselves proliferate and provide protein to the animal.
Yeah.
Moreover, the indigenous grasses are producing more methane, which is a problem. And that's why even now we have the weather is really haphazard. So we are looking at something that can preserve the fragile ecosystem in the rangelands, at the same time minimize global warming. And so as a lay person like myself, I would still go for these indigenous grasses. I'm not very persuaded, although the data is here. I would still go for the indigenous grasses for the simple reason that they persist, they provide feed when there's no rain, and they're producing less methane emission.
Yeah.
So what I'm suggesting is that you really need to go back to your conclusion and bring it out strongly, where you would recommend the introduced grasses that require more moisture, more fertilizer in order for them to produce. And also now that are going to give us more methane, what can we do to reduce that methane? On the other hand, you look at the indigenous grasses and say, yes, they are low in crude protein, but perhaps if we get another legume, something that is also hard in the ecosystem that can combine with that indigenous grass and be able to provide feed for the animals. Have you gotten my explanation, Anastasia?
Yes, yes.
Okay, are you going to think through it?
Yes.
Okay, thank you. Any other comment or a question for Anastasia? So if you are all satisfied, we thank you so much, Anastasia, for your presentation. We wish you well in your future endeavors.
Thank you.
And I just want to let the participants know that the other persons listed on the program are not available. They have not sent their presentations. They didn't send before. They haven't sent this day as we have tried to look for them. So apparently this is the end of this session. So please do not exit the meeting. You can exit the room and join any other session, and then you will be given the poll to respond to from whichever session you have joined. So thank you so much for participating. Thank you.